Hi everyone. In the last lecture, we have seen how transcription is initiated in E. coli and how the mRNA synthesis is elongated, that is elongation of transcription in E. coli. In this lecture, we will continue with the transcription in bacteria and we will see how transcription is terminated in bacteria, that is termination of transcription in bacteria. Termination of transcription or RNA chains occurs when RNA polymerase encounters a termination signal. And when RNA polymerase enzyme encounters a termination signal, the transcription complex, this RNA polymerase enzyme dissociates from the DNA, releasing the nascent RNA molecule, which is the transcript. There are two types of transcription terminators in E. coli. The first one is called as row-dependent terminators, which involves the presence of a protein called as row protein. That's why it's called as row-dependent terminators. And the mechanism is called as row-dependent termination mechanism of transcription. And the second one is called as row-independent terminator because it does not involve row protein or any other protein. And the mechanism of transcription is called as row-independent termination mechanism. We are going to see both of the terminators and the termination mechanisms in detail in this lecture. We'll begin first with row-independent terminators, that is row-independent termination mechanism. So this is the terminator sequence. First of all, we need to understand that this is a DNA sequence. And it is a terminator sequence. Now, what do I mean by a terminator sequence? This is part of the gene, yes. In one of the previous lectures, we have seen that a gene can be divided into three parts when it comes to transcription: the promoter sequence, the RNA coding sequence, and the terminator sequence. And we have seen this diagram. So this is the promoter sequence, which is involved in initiation, where the RNA polymerase orients itself and it's upstream of the coding sequence. This is the coding sequence. And this parrot green color is the terminator sequence. In this lecture, we are talking about this termination or terminator sequence. And this parrot green color terminator sequence is shown in detail here. In row-independent terminators, they have a GC-rich sequence. So when we say a GC-rich sequence, see the sequence from here till here, it is rich in GC. So there are some TA base pairs, but otherwise you can see it's full of GC base pairs. Again, there's a AT base pair, but again GC. There are two AT base pairs, again GC base pairs. Again, there is one AT base pair here and one here, but this is full of GC base pairs. So this sequence from this region to this region is GC rich, and this GC rich sequence is followed by 6 or more AT base pairs with ACE in the template. So here where the GC rich sequence ends, you can see there are 6 or more AT base pairs with ACE in the template strands. So far what we have seen in this row independent terminators, there's a GC rich sequence which is followed by 6 or more AT base pairs with ACE in the template strand. Now let's see more about this GC rich sequence. This GC rich sequence has inverted repeats. What do you mean by inverted repeats? The inverted repeat regions are the two shaded regions. One is this and second one is this. And these inverted repeat regions are inverted. What do you mean by inverted? Now let's see. Let's start reading this sequence from, let's say, this end. So the sequence here, if I read is T, G, double C, G, double C, and so on. And if I've read this sequence T, G, double C, G, double C in this direction. In this another shaded region, if I read the sequence in the opposite direction in the lower template strand, it's same. T, G, C, C, G, double C. So this time I have read it in this direction. So the sequence is same, but it's inverted. And that's why it's called as inverted repeat. So it's in opposite direction. This shaded region we read from this way, and this shaded region we read from this way. So it's inverted and it's repeated. So it's called an inverted repeat. So they are not only inverted, since they are repeated, they are complementary to each other. So let's see what happens. So the RNA polymerase holoenzyme has bound to the promoter -10, -35 sequence. And then from the +1, the mRNA synthesis has begun. And the mRNA synthesis proceeds through the entire coding sequence. And when the RNA polymerase reaches this terminator sequence, here also denaturation occurs, and this GC-rich sequence will be copied into mRNA. So transcription, when this GC-rich sequence will be copied into mRNA, can I say transcription of the inverted repeats have happened? Yes. And when the transcription of the inverted repeats happened, in this step, you can see that this first shaded region, part of the inverted repeat, has been copied into mRNA here. And the second shaded region, this one, is being copied now. And once these inverted repeats are copied into mRNA, do not forget that they are complementary, they are inverted. So once the mRNA is being made, there would be a rapid folding of the mRNA into a hairpin-like structure, and this would happen immediately after the mRNA synthesized. Now, what do you mean by hairpin-like structure? So in this step, till this step, you can see that this mRNA is single-stranded, and these inverted repeats have been copied. Immediately after the synthesis, in the next step, you can see that these inverted repeat regions in the mRNA will form base pair with each other. And because these inverted repeats were complementary to each other, they can form base pair. And once they form base pair, you will have a double-stranded region. Yes, the mRNA is single-stranded, but a double-stranded region will form due to this intramolecular hydrogen bonding. And this double-stranded region has formed because of the hydrogen bonding between the inverted regions which were complementary, and this resembles like a hairpin. That's why it's called as a hairpin structure. What next? I said that after these inverted repeats in the DNA, there is a 6 or more AT base pairs. Yes. With A's in the template strand. Yes. And if there are A's in the template strand, in the mRNA, there would be uracils, a string of uracil residues, because there is no thymine in the mRNA. So the adenines in the template strand give these uracils in the mRNA. And because of this hairpin structure, RNA polymerase has halted. It has come to a pause, like how a video game is paused. So there is pause in the chain extension because the RNA polymerase has paused because of this hairpin. And since the RNA polymerase has paused, now the mRNA can be dissociated, and this AU base pairing is also weak. It's an area of weak hydrogen bonding, and it's easier to break the 2 hydrogen bonds between adenine and uracil rather than 3 hydrogen bonds between guanine and cytosine. And since this AU base pairing is weak, the transcript is released. And this transcript is released not just because of the weak AU base pairing, but also RNA polymerase has paused here. And RNA polymerase has paused here because of this hairpin structure. This hairpin formation occurred because of the inverted repeats. So this is how termination of transcription in E. coli occurs via row independent mechanism. And these rho independent terminators have GC rich region, which have inverted repeats, followed by 6 or more AT base pairs with ACE in the template. And the hairpin possesses the RNA polymerase, and the weak AU base pairing releases the chain. Now we will see the other type of the second type of termination mechanism, which is rho dependent termination mechanism. Now, certain genes in E. coli have the terminator region, which is not rho independent terminator, but rho dependent terminator. So if the gene has rho dependent terminator, it's not going to be terminated by the mechanism we have just discussed. This rho dependent terminator, the transcription will be terminated via rho dependent termination mechanism. The mechanism by which rho dependent termination of transcription occurs is similar to rho independent termination in that both involve formation of hydrogen bonded hairpin structure upstream from the site of termination. So as the name suggests, rho dependent, we may see that there are many differences between rho independent and rho dependent termination mechanisms. Yes, there are differences. One of the major differences that rho dependent mechanism is going to involve rho protein, which is not there in rho independent mechanism, but there are some similarities as well. And one of the major similarity is that rho dependent termination mechanism, like rho independent termination mechanism, also involves the formation of a hairpin structure upstream from the site of the termination or before the site of termination. So if there's hairpin, what else is common? And in rho dependent termination also, like rho independent termination mechanism, the hairpin will impede the movement of RNA polymerase, causing it to pause. So here also there will be hairpin formation. Here also the RNA polymerase will come to a halt or will pause because of this hairpin structure. But what next? However, rho-dependent terminators contain two additional sequences. A 50 to 90 nucleotide sequence, which is upstream from the inverted repeats which give rise to the hairpin. And this 50 to 90 nucleotide pair sequence not only is upstream from the inverted repeat, but produces an RNA strand with many cytosines, but very few guanines, which forms no hairpin or other secondary structure. So when I said no hairpin or other secondary structure, just now we have said hairpin structure is formed in rho-dependent mechanism. We mean that there is no hairpin in this region, which is that 50 to 90 nucleotide pair sequence. And the second additional sequence is a sequence specifying a rho protein binding site called RUT sequence. This is called as RUT sequence. This is for rho utilization site. And this RUT sequence is a sequence for rho protein binding site, where the rho protein will come and actually bind to this RUT sequence. And rho protein is the main player in this mechanism. Near the 3' end of the transcript, this sequence is there. So what happens? Rho protein comes and binds to this RUT sequence, and rho protein not only binds to the RUT sequence, but will move in the 5' to 3' direction. It will run in the 5' to 3' direction. So let's say that this is the rho protein and it is bound to this RUT sequence. And by binding to the RUT sequence, or after binding to RUT sequence, this rho protein will move or will run in 5' to 3' direction. So after some time, it will come here. After some time, it will come here. And it keep on moving in 5' to 3' direction. And it is chasing RNA polymerase, which is ahead. So let's say this is RNA polymerase much ahead. And this is rho protein, which is behind. So rho is chasing RNA polymerase. And you would say that if rho is chasing RNA polymerase is moving also. So rho will never be able to chase RNA polymerase, but it's not like that. Because of the hairpin structure, the RNA polymerase has paused. And now Rho will be able to chase the RNA polymerase. And as I said, when RNA polymerase encounters the hairpin, it pauses, allowing Rho to catch up. And when Rho reaches there near the hairpin, Rho is nothing but an helicase enzyme. Helicase has the ability to unwind 2D strands, whether they are DNA, or DNA, RNA. In this case, Rho unwinds the DNA RNA hybrid at the terminus and releases the RNA transcript. And the termination of transcription is achieved. So this was Rho dependent termination mechanism, where there is involvement of Rho protein, which binds to the Rut site, moves in the 5' to 3' direction, chases the RNA polymerase, and RNA polymerase, which has paused because of the hairpin structure, which is similarity to Rho independent mechanism. This allows Rho to catch up, and using it helicases, it unbinds and releases the nascent RNA transcript. So in this lecture, we have seen two major mechanisms of termination of transcription in bacteria: Rho independent termination mechanism and Rho dependent termination mechanism.